We'll start now. So welcome to your first credit preparation. This is Hematology. I hope you guys have started studying. Okay, nice. Okay, so we'll slowly start with the structure of the test. We have discussed this in the introduction seminar as well. There will be 30 questions and you will have a time limit of 20 minutes, all MCQs, and you may have more than one correct answer. But remember, there are also negative marking, so unless you're completely sure of the answers, like don't pick everything, yeah? Pick the answers you're most sure about. Okay, so now that we're clear about that, let's start with the seminar. Okay, so we begin with homeostasis. Now, homeostasis is important for all your body systems. It is the ability of the organism to maintain the stable internal environment despite changes in the external environment. And you have this question in your PPT. Why is it so important? Now, these are your reasons. Maintain stable internal conditions like temperature, pH, all those things. So it's for maintaining the internal condition, ensuring proper enzyme activity. All your enzymes, they have a specific requirement, temperature or pH to work. So homeostasis is once again responsible for keeping that, supporting normal cell function, survival in changing environments, maintaining energy balance, that is your glucose and oxygen intake, are all controlled by homeostasis. Okay, so now we move on to the next one, which is body fluids. Now, body fluids and homeostasis, they go hand in hand. Body fluids, what are they? They are the aqueous solutions which are containing both inorganic and organic substances. Okay, like ions, glucose, proteins, etc. And if we take a young healthy man who has a body weight of around, let's say, 70 kg, the total body fluid of that person would be 60% of his body weight. So by simple calculation, we can say it's around 42 liters of his body weight. Okay, and the main factors affecting this is age. So older people, they have more body fat, correct? That's why their body fluids will be less. Then gender, women once again have more body fat. That's why their fluid content will be less. And also once again percentage fat, right? So body fluids are mainly divided into two broad categories, the intracellular fluid and extracellular fluid. Once again, this is taken from your PPT, so make sure to learn this diagram very well, it is quite important. You need to know the values of all of these fluids. So ICF is making up two-thirds of your total body fluid. External is making up 1/3 of your total body fluid, which is further divided into interstitial, intravascular, and transcellular. Anyone can give example of transcellular fluids? Yes, cerebrospinal fluid is an example. It's making up 1 to 2.5. It's not very important, but these two are very important, interstitial and intravascular. Alright? So we can go to the next one. Measurement of the body fluid compartments. Now this is a very summarized way of studying, like I've created this table for you. So the main formula is this, the dilution principle. V is equal to N by C. It's quite important. And the basic principle of this is that you introduce an indicator into the body and then you measure the presence of that indicator. And that's how you calculate the volume of the body fluid. You measure the concentration of the indicator. The most common indicators used, you can see here, these, okay, this is not working. These are the most common indicators used, very, very important. You can get like any question about this, like what are the indicators used for total body fluid, okay? These are the ones, and just remember that intracellular fluid cannot be measured by an indicator. It can only be measured by this formula. Intracellular fluid is equal to total body fluid minus the extracellular fluid. Okay? So that's the only one which is not measured by the indicators. And just have a look at the requirements for the indicator as well. This is also important. It can come in the exam once you pick the main requirements for the indicator. And also, I want to add side note, this is important for the final. Like, they can ask anything from here. So that is about it for the body fluid parts. We'll move on to the next topic.
working normal flow. And think of it as the body's emergency plumbing system. So when a vessel breaks, we need a quick seal, but not a permanent blockage. And there are four main stages for hemostasis. First being vascular reaction. The damaged vessel constricts, and that reduces the blood flow. Then we have platelet blood formation, where the platelets arrive first, and they kind of seal up the damaged lining. And then you have coagulation, where the fibrin threads, they act as a mesh to stabilize the blood. And then we have fibrinolysis. So once the repair is done, the clot has to dissolve. And throughout this, the body keeps a balance, because if there's too little clotting, it could lead to bleeding, and if it's too much, it could lead to thrombosis. Okay, so let's start with what happens the moment a blood vessel is injured. The process that stops bleeding but also stays controlled to prevent overclotting, and this is known as the hemostatic response. So our first thing that happens is the vascular reaction, and the first reaction is an instant local vasoconstriction. So I'll just draw something for you guys to understand better. So this is your endothelial lining, and here is your collagen, and here you'll find your smooth muscle cells. So when your endothelial lining damages, right, over here, the smooth muscle, it gets, so the lining itself, it releases some chemicals like endothelin, serotonin, and those guys will go and act on the receptors that are on your smooth muscles, and these smooth muscles will cause vasoconstriction. And that vasoconstriction is basically the smooth muscles constricting so that the damage is reduced. And then, so that's your first step. And there's many mechanisms that happen here. One is myogenic reflex, which is when the smooth muscles constrict, like I showed. Then you have your neural reflex. That's when these, so there's also pain receptors that are along there with the smooth muscles, and that's also where the noisy receptors act. And then you have your humoral factors like serotonin or thromboxane, which I'll explain, and they further constrict the vessel. And this narrowing gives platelets a chance to arrive and plug the gap before too much blood escapes. Then we have our platelet plug formation. So this is your primary hemostasis, and this is where the platelets perform a temporary plug. So again, I'll show you guys what happens exactly. So I hope everyone can see. So again, you have here your endothelial lining, then you have your collagen, and then you have your smooth muscles. So again, when your endothelial lining is damaged here, the collagen is now exposed. And when the collagen is exposed, the endothelial lining, which is not damaged, it releases something which is known as the 1 milligram factor. And this is basically a chemical that is kind of activated here. So you have the 1 milligram factor that shows up. And what this guy does is, so it will kind of like attract the platelets that are around, and the platelets have a very specific receptor, which is called the GP1B receptor. And this 1 milligram factor, what it does is that it will connect to the GP1B receptor of the platelets and kind of drag them down to the damaged endothelial line. And then this whole thing will release some chemicals, and those are your thromboxane A2, your serotonin, and also your ADP. So then what they do is they will attract more platelets, and more platelets will come here, and they'll all get accumulated here, and they'll form a platelet plug, and that's the formation. Then aggregation is basically, now your GP1B has come and made the platelet plug. There's also another receptor on the platelet, which is called the GP2B3A. It's a bit hard to remember, but like GP2B3A, and what it does is that it will hold the fibrinogen in between it. So this is one platelet, this is one platelet, like this, and together they will combine and they're held together, and that's your aggregation. So that is basically your primary platelet plug formation, and it can, too much clotting, like I said, is also because it can cause thrombosis. So for that, we have something known as prostacyclin and nitric oxide that's also released by the endothelial lining. And these guys act as anticoagulants. Basically, they make sure that there's not too much clotting happening. And also, there's a clinical note, aspirin. Aspirin works right here. It irreversibly inhibits cyclooxygenase, which is responsible for making thromboxane A2, which is a primary coagulant. Okay, so think of homeostasis like an emergency team arriving at a crash site. So there are two roads that will lead to the scene, the intrinsic and the extrinsic, and they both will meet at the same intersection, which is your common pathway, which is factor 10. So this is where they will come and try to stop the bleeding. So you have your extrinsic pathway. Your extrinsic pathway is the fast route or the express route, and this is triggered by tissue trauma at the external side. So the outside, at the blood vessels, that's if your damage happens, there's something known as tissue factor, which is initiated, and this tissue factor also is known as factor 3, and this is released by damaged tissue. Now this factor 3, if you look here, extrinsic, tissue factor 3 will bind with factor 7, and they both will form a complex. And these two factors together will go and activate factor 10. And factor 10, like I said, is in your common pathway. And this one is a simple chain. It's fast, but it's short-lived, so it's like a quick alarm response. Then you have your intrinsic pathway. Intrinsic pathway over there is the slower route, and this is known as the internal maintenance team. And this one is triggered by blood trauma, or if it comes in contact with collagen, like I showed there, if the endothelial lining is damaged or collagen is exposed. And then what activates is factor 12. Now, how does that activate? Through auto-activation. Nothing really activates it. It just realizes that there's damage, so it has to activate. Then factor 12 will go and activate factor 11. Factor 11 will go and activate factor 9, and factor 9 will go activate factor 8. I know this sounds crazy, but just try to follow with me, so it'll be fine. And when I say these clotting factors, I'm talking about a group of special proteins that are made by the liver that circulate in our blood in an inactive form. And when a vessel is injured, these factors get activated, and like a domino effect, they form the fibrin mesh. And so I'll show you guys mnemonic as well to remember, but I'll first talk about the common pathway. So the common pathway, now both extrinsic and intrinsic together have come to form and activate factor 10. And factor 10, once it's activated, along with factor 5, calcium and phospholipids, all of them together will go and activate factor 2. Now what is factor 2? Factor 2 is basically prothrombin, and that will get activated to thrombin. These words might be familiar, prothrombin, thrombin, you must have heard in A level. So prothrombin is basically your factor 2, and thrombin is your factor 2 that's activated. And then, once that happens, factor 1, which is your fibrinogen, again must have heard of it, is activated and forms fibrin. And like we said before, fibrin is the mesh that's made, and hence your clot finally is made. So I know these factors, I said like way too many numbers, so it must have been hard to follow, but a quick way to remember is these factors. So just remember your common pathway factor is factor 10, yeah? So here is your factor 10. And extrinsic pathway, I said factor 3 is activated, and then your factor 7 is activated. So 3 plus 7 is 10. Then intrinsic pathway, I said factor 12 activates, then factor 11 activates, then we can skip 10 because we know it's the common pathway, and then factor 9, and then factor 8, and then factor 10. That's how you remember the intrinsic. And then over here you have factor 5, factor 2, and factor 1. So just remember 5 into 2 is 10, 10 into 1 is 10. So these all, it's just a way to remember all these factors because it's too much is going on. And also remember factor 2 is your thrombin and factor 1 is your fibrin. So now that the clot is formed, we need to talk about something else, and that is how does the blood keep itself naturally thin? There's not always going to be a clot forming, right? It's usually the normal thing of blood is to remain thin. So how does it do that? So, okay, so we have a bunch of chemicals that are present in our blood. One is antithrombin 3. Now this is the main inhibitor. It neutralizes thrombin, the one that is responsible of converting fibrinogen to fibrin. And it also inactivates factors like 9, 10, and 11. And think of this as body's internal heparin. Heparin is clinically a drug you give, and this enhances antithrombin 3 activity, so it acts fast. So antithrombin 3 is your main anticoagulant. Then we have protein C and protein S. Protein C and protein S are activated, and these guys will also inactivate factor 5 and factor 8. And yeah. And then you also have nitric oxide. Nitric oxide is naturally released by the lining, by the endothelial lining, and it's one of its roles is to make sure that there's no aggregation happening, and like it prevents platelet aggregation. And the key idea is that coagulation and anticoagulation, they work like a seesaw. So the goal is balance and not zero clotting. Finally, fibrinolysis. Once the vessel is repaired and the body has to remove the clot, that's fibrinolysis. And it starts with something known as plasminogen, which is an inactive protein bound to the clot, and that is activated into plasmin by something known as tissue plasminogen activator, TPA, or also urokinase. Plasmin what it'll do is now it'll digest the fibrin, the clot we made, into smaller soluble pieces, and that's called your fibrin degradation products. And this will again reopen the vessel. And so that's basically a gist of your fibrinolysis. And the summary is that the clot formation will stop the bleeding, the clot inhibition will prevent excess of clotting, and fibrinolysis will clean up once the healing is done. And yeah, that's hemostasis. Okay,
so guys, I'm going to be explaining functions of blood. Functions of blood, the most basic function is transport of gases. So it transports oxygen, carbon dioxide, and other gases from the tissues to the lungs, and the lungs to the tissues. Then we have maintenance of homeostasis, which was explained by her earlier, and that's pH balance, osmotic balance. Osmotic balance is really important because blood carries plasma proteins, and these plasma proteins are responsible for pulling the water into the vessels and not letting them pool up in the interstitial tissues, or else the tissues get edematous. Then we have hemostasis, which was just explained, and then humoral control. So blood also is responsible for carrying hormones, which help us conduct many important processes in our body. Then there are immune functions. Immune functions basically when there's inflammation happening at a certain site, it carries immune cells like neutrophils towards it, or it carries antibodies, and there's a complement system that you guys will study in immuno, which is really important too, and blood helps carry it. And then thermoregulation. So blood also helps maintain the temperature level, and this can be done with the help of vasodilation or vasoconstriction. So vasodilation is basically when the vessels they dilate, and this happens when the internal body temperature has gotten too hot. Because the body is too hot, it needs to let the blood or like the fluid evaporate and promote evaporative cooling. So that's the reason vasodilation happens in heat, and in cold environments you get vasoconstriction to conserve heat. Okay, so RBCs. RBCs are really specialized cells that are enucleate, and they don't have mitochondria or any organelles. This is to create more space for hemoglobin. They have a biconcave shape with an indent, as you can see in the picture. So this indent helps it become flexible, and it also helps it squeeze through capillaries, and this avoids rupture. Then the origin and the production of RBCs. So RBCs are produced in the bone marrow. but it's not always like that. In the fetal period, in the first trimester, RBCs are produced in the yolk sac. In the second, the liver takes over, and in the third, we have bone marrow. And RBC production is stimulated by erythropoietin. Erythropoietin is a hormone that is produced by kidneys, around 90%, and partly by the liver, and it is also produced by the liver as like a major organ during the fetal period. This is not very important, but I was asked a question on it, so I'm just putting it out there. And then the lifespan is 120 days, and the composition. Yeah, so the only thing that you need to remember in the composition is that we have spectrin and anthiorin, which help with the flexibility of the membrane, and the membrane also carries antigens ABO, which determine the blood group. Yeah. So metabolism of RBCs. So metabolism is not just how it gets its energy, it also means like the sum total of chemical processes in the RBC. So you have the energy source, obviously the most important one. It only uses glucose for energy, which enters the RBC through GLUT1 channels. Now you don't have to know this in detail, but just remember GLUT1 channels. And what happens is they can only be processed by anaerobic glycolysis. Why? Because they don't have mitochondria. So that's very important. Then the hexose monophosphate shunt. You just need to remember that it reduces NADPH for antioxidant defense because RBCs are involved in oxidative, not oxidative processes, but they're around oxygen a lot, so that can cause oxygen poisoning sometimes. So to prevent that, you have the antioxidant defense. Then 2,3-DPG. 2,3-DPG is a very important compound, and it's basically produced by the endothelium and the RBCs also. What it does is this is just for you guys to understand, you don't have to know this in detail, but what it does is it will go and bind onto hemoglobin where oxygen doesn't bind, so it's a different side, and it makes hemoglobin have less affinity for oxygen. So since it has less affinity for oxygen, it lets go of oxygen, and by letting go of oxygen, the tissues get the oxygen. So this would be especially needed in oxygen, and 2,3-DPG levels also rise when you have hypoxic conditions in the body. So since the tissues are not getting enough enough oxygen, you have 2,3-DPG increasing. Then ATPases are basically ion pumps or channels kind of, which use ATP, and they are required to keep a balance of ions. So this is very important because say calcium for example, if that increases in RBCs, what happens is the flexibility of the RBC decreases. So you can see this in old RBCs where they're not flexible anymore and they start rupturing in your capillaries. Then we have the membrane permeability, so it's permeable to gases, water, bicarbonates, and chlorine. These bicarbonates and chlorine are very important, and I'll be explaining that soon. Then we have the antioxidant systems. So methemoglobin reductase is one of the most important antioxidant systems. So what happens is, in general, in random processes that occur in the body, the RBCs that carry hemoglobin, which have iron in it, in the Fe2 plus form, converts to Fe3 plus. It's a very spontaneous process, and it's a result of many random processes in your body. So Fe3 plus form of iron cannot bind to oxygen. Since Fe3 plus form of iron cannot bind to oxygen, if you get methemohemoglobin, which is basically hemoglobin with Fe3 plus, oxygen cannot bind and your tissues can start becoming hypoxic. So to avoid this, we have an enzyme called methemoglobin reductase, which converts Fe3 plus back to Fe2 plus whenever it gets oxidized, and this helps oxygen bind. Glutathione is also another antioxidant system, and it works similarly. Then carbonic anhydrase system, and she's going to be explaining it soon. So yeah. So hemoglobin. These two terms, chromoprotein and metalloprotein are kind of important because when we're talking about hemoglobin in your credit test, they might just refer to it as a chromoprotein or metalloprotein, so don't get confused. Chromo means color and metallo means metal. So the hemoglobin protein has metal and color being the red pigment and metal being iron. And the structure, it has 4 subunits to identical heterodimers. Heterodimers are basically dimers, as in two things joined together, but they're not the same things. So that's the reason it's heterodimer. So 2 alpha chains and 2 beta chains, and then heme. Heme is heme can be bound to two different types of chains. So based on that, we know what form it is. So HbA, which is mostly in like the major adult form, has 2 alpha chains and 2 beta chains. HbA2, which is less than 3% common, it's quite a rare type of hemoglobin that has 2 alpha chains and 2 delta chains. And HbF, this is one of the very important types, which is 2 alpha chains and 2 gamma chains. This kind of hemoglobin has much more affinity for oxygen, so that's all you guys need to know for that. These values are important, so please memorize them. And the basic functions are transport of oxygen, transport of carbon dioxide, and then maintenance of blood pH. Again, I'm just going to explain that. Okay, so HV derivatives. HV derivatives basically come from different gases binding to hemoglobin. So firstly, we have oxyhemoglobin, which is the most important one, which is oxygen binding to Fe2+, as I mentioned earlier. One hemoglobin binds to 4 molecules of oxygen. So this happens because hemoglobin has 4 subunits, so it'll have 4 ions in Fe2+ form, and that's how it binds to 4 oxygen molecules. Then 1 gram of hemoglobin carries 1.34 ml of O2. These are very important values, please don't skip them. And the affinity for oxygen is affected by partial pressure of O2, partial pressure of carbon dioxide, pH, temperature, 2,3 dpg. So now, if you guys remember what I said about 2,3 dpg earlier, then you can look at the graph on the side. They're talking about how 2,3 dpg increases on the right side. So that means oxygen affinity is going to decrease. So along with that, if carbon dioxide increases, then the affinity decreases for oxygen, and so on. So left side is when oxygen affinity is more, and right side is when oxygen affinity is less. If you don't understand this graph, please feel free to ask me questions. Then we have deoxyhemoglobin. Deoxyhemoglobin just means that the hemoglobin doesn't have oxygen, so it's not bound to anything at this point. When it's not bound to anything, it can bind to H+, which means it acts as a buffer, so it can temporarily get rid of H+ in your blood when there is too much acid in your blood, which is known as acidosis. Then we have carbaminohemoglobin. So notice how it says carbamino. That's because carbon dioxide binds to the NH2 group of the globin. So it's not binding to Fe2+, it's binding to the NH2 group, which is the amino group. And it lowers O2 affinity and facilitates O2 release. So there's this thing called allosteric effect that comes into play here. Allosteric effect is basically, since there are 4 subunits, if one subunit binds to oxygen, then the rest 3 are more likely to take up oxygen. And the same way, if one subunit loses oxygen because carbon dioxide binds to it, then the rest of the subunits are more likely to leave it. So it's just something that helps oxygen release and binding much easier at different parts of the body, like the lungs and the tissues. Then carboxyhemoglobin. So this is basically carbon monoxide. Carbon monoxide binds to oxygen in the same place that binds to hemoglobin in the same place that oxygen does. And carbon monoxide has 200 to 300 times the affinity. Since it has this much affinity, once hemoglobin attaches to carbon monoxide, monoxide, it cannot let go of it, and that creates the problem of carbon monoxide poisoning, and that's the reason it's really dangerous. And the skin starts turning pink and red. Then methemoglobin. Methemoglobin is the same thing that I told you guys about methemoglobin reductase. So when Fe2 plus converts to Fe3 plus, here we need the enzyme methemoglobin reductase. But if it's not present and your RBC stay in the methemoglobin form till the end of time, then you get this disease called methemoglobinemia, methemoglobinemia.
We're going to be talking about acid and base balance. So our body is obsessed with our body is obsessed with trying to keep everything in balance, and especially pH. So our normal blood pH is always between 7.35 to 7.45, and very important, remember that value, engrave in your head because it comes up a lot of times. And if it ever goes even slightly below 7.36, you would call that acidosis. And slightly above 7.45, you would call it alkalosis. And so calculating pH on its own, it's a very minuscule number. So we use the formula pH equals to minus log of H. And H in the brackets basically means concentration of the hydrogen ions. So that's that. And to keep the pH steady, three systems work together. So you have your buffers. Buffers, they act instantly, and they will quickly start working their system to fix the balance. And that they will mainly through the bicarbonate buffer. I'll explain the buffers in a second. And you have your lungs. Lungs will respond within minutes, and they will, what they'll do is they'll try to adjust the carbon dioxide, and our carbon dioxide is basically the base in our body, the acid in our body. And then you have kidneys. Kidneys are slower. They control the bicarbonate ion at CO3 minus, and that acts as your base. And in short, buffers are your firefighters, lungs are your ventilators, and your kidneys are your long-term pH managers. So now we'll talk about the buffers. Buffer systems. So now we'll look at how the body actually reacts to sudden pH changes. The most important out of all of them is your carbonic acid buffer, and this one amounts up to 53% of your buffer capacity, so it's very important. And how this works, guys, remember this equation really well, because this will make it so much more easier, because you see that this is a reversible reaction, so it can move either to the left or right depending on what's more, either your acid or your base. So when your acid increases, meaning your hydrogen ion concentration increases, there's too much of acid in our blood. So we don't want that, we want it to be equal. So what will happen is, the H will react with the bicarbonate ion at CO3 minus, and then H2CO3 is made, and that's your carbonic acid. Carbonic acid will then immediately break down to water and carbon dioxide, and carbon dioxide is removed via the lungs by exhalation. And that is if there's too much H plus. Now what if there's too much base? What if there's too much hydroxide ion? What will happen is that when your hydroxide ion is increased, this will react with the bicarbonic acid, H2CO3, and then form water and bicarbonate ion. So basically what I'm trying to say is that when your base increases, it will move to the left to release more hydrogen ions. And if your acid increases, meaning your hydrogen ions increases, it'll move to the right to form bicarbonic acid. So they're just working all the time to make sure that your blood pH remains normal. And like I said, 53% of the total buffer capacity, and it also depends on the ratio, the effectiveness. So the bicarbonate and the carbonic acid are always in the ratio of 20:1. Then we have our phosphate buffer. The phosphate buffer works mainly inside the cells and in the kidney tubules, and it's good for fine-tuning pH when you have urine formation. And they don't go a lot of detail into it, but then you can read the lecture for more detail. And then you have plasma proteins. Plasma proteins, especially histidines. Now, where are these histidines found? In the hemoglobin. And these histidine residues, they help, but they're less effective because their PK is not that close to the blood. What is PK? I forgot to mention. PK is the value which shows how easily the acid will lose the hydrogen ions. So if PK is low, meaning it can readily, like the acid can give away its hydrogen ions. So in that sense, plasma proteins, even though they have buffering capacity, they're not that, because they only make up 7% of the total capacity. Now you have your hemoglobin. So Shah explained hemoglobin really well, but you also should know that the hemoglobin also has a buffering role. So it also acts as a buffer. How? Now, there's something known as Haldane effect. So hemoglobin, like I said, has histidine groups, and these histidine groups can accept or donate hydrogen ions depending on the pH level. So we know what hemoglobin's main role is, right? And that is to collect oxygen and transport it to your tissues. So when you're in the tissues, the hemoglobin is giving up. So in the tissues where oxygen is low, right? The deoxyhemoglobin, which is the empty hemoglobin, which is not carrying any oxygen, it grabs onto the hydrogen and carbon dioxide. So in the tissues, you have less oxygen, right? So that time the hemoglobin is kind of free, and when it's free, it can bind onto H plus and carbon dioxide. And then, similarly, in the lungs is where you have high amounts of oxygen, right? So that's where your hemoglobin is kind of occupied, known as oxyhemoglobin, because it's holding onto the oxygen. So it will give off hydrogen ions and carbon dioxide. So in that way, this whole process, they keep balancing between giving up oxygen, taking in carbon dioxide and hydrogen ions, and vice versa. And this whole thing is known as the Haldane effect. So the hemoglobin dissociation curve. You guys, this table is so important. Like you have to remember it very well. And Haldane effect is basically when it shifts to the left, right? Haldane effect. It's to the left, right? Yeah. Okay. So this whole process is 35% of the total buffer capacity. So a very important part of acid base. Now disorders, they will ask you these disorders in your final and even in your credit. So they're very important. Now there's 4 main disorders, 2 respiratory and 2 metabolic. So first I'll talk about respiratory acidosis. So respiratory acidosis, again, acidosis means when your pH levels are low. So respiratory acidosis, how does it happen? By hypoventilation. Hypoventilation is when you're not exhaling enough. Your breathing is very low. And you're not exhaling carbon dioxide, and this will build up the carbonic acid. If you remember the equation from before, if you have more carbon dioxide, it can go and form carbonic acid. And then this carbonic acid will make the pH drop, so that will lead to acidosis. But how is this maintained? What does the body do in return? It tells the kidneys to start working. What the kidneys will do is they'll retain their bicarbonate ion at CO3 minus. And since that is your base, kidneys will try to hold on to that and make sure that it's balanced. Then you have your respiratory alkalosis. Respiratory alkalosis, complete opposite of acidosis. This is when you're hyperventilating, especially during anxiety or when you're at a high altitude. You're trying to get rid of the carbon dioxide and get more oxygen. So at that time, you go into a respiratory alkalosis because you're blowing off too much carbon dioxide. You're getting rid of too much acid. And because of that, the pH will rise and that will lead to alkalosis. But again, how does the body respond to this? It'll again allow the kidneys. The kidneys will, what they'll do now, opposite of what they did in acidosis, they will try to get rid of the bicarbonate ion because that is your base. And you already have way too much base in your body, so you need to bring that balance back to normal. So that is your respiratory. Respiratory, like I said before, lungs. Lungs' main role is to deal with the partial pressure of your carbon dioxide, either by hyperventilation, hypoventilation. Then you have your metabolic acidosis and alkalosis. Metabolic acidosis is when you have too much acid or too little bicarbonate ion. And that is like enduring diarrhea. When you have diarrhea, you give away a lot of your like a lot of substances that are high in your pH. So you blow off too much alkaline stuff, so your base. So you have too much, your body's in metabolic acidosis. And even in diabetic ketoacidosis, which you will learn later. So what will the body do now? The body will now compensate for this via the lungs by hyperventilating, and that is known as your small breathing, which is when you're breathing really fast to get rid of the carbon dioxide because your body is in a state of acidosis. Finally, you have metabolic alkalosis. Metabolic alkalosis happens when there's too much base, often from vomiting or diuretics. So when we vomit, we're getting rid of the substances in our gastric juice, which are high in acid. They're very acidic. So when you vomit, your body goes into a state of metabolic alkalosis. And how will the body compensate? By telling the lungs to hyperventilate. So that will retain the carbon dioxide to make more acid. So acid-base balance is just the role of kidneys and lungs kind of coming together to compensate for each other's loss, whether it's acid or alkaline. Now, parameters.
By the way, guys, for small breathing, it's rapid but also very, very deep. So it's not just like tachypnea. Yeah. So the physical parameters, unfortunately, guys, you have to remember all the values. But if you remember the logic behind it, then it will get easier. Try to write it on one page and study it for credit. So RBC count. RBC count, as you can see here, males have a higher RBC count than the females, and this is because males, they have more RBCs in general. Now this happens because obviously they're bigger, their bodies are bigger, and also they have testosterone. Testosterone is a stimulator of erythropoietin, which stimulates erythropoiesis, so that's the reason men have more RBCs. Then newborns, they have more RBCs too, but this is a different, there's a different reason, which is growth and development. So they need a lot of RBCs for development. And along with that, newborns have smaller bodies, so like by the ratio also they have a lot of RBCs. Then the changes in count. So changes in count can be of two types, that is absolute and relative. Absolute is where there is actually a rise or a fall in the number of RBCs. So you get conditions like polycythemia in say altitude. So in high altitude, since not enough oxygen is reaching, you have to, like the body has to create more RBCs so that more oxygen reaches the tissues. And erythrocytopenia, which is I think also known as oligocythemia. So erythrocytopenia is basically in anemia, blood loss, where you're actually losing RBCs. In anemia, why you lose RBCs is that when, say for sickle cell anemia, you have a different kind of RBC. So it's like sickle cell, the shape is known as drepanocytes. So the body identifies it as a foreigner body, and that's the reason it destroys it. So anemia is also one of the examples of erythrocytopenia. And then there is a relative change in count. So relative change in count is basically, it just looks like the RBCs have increased or decreased, but actually nothing happens. So it's the plasma that's changing. So either hemodilution, so you've added like saline to the plasma, in an infusion. So that would show a relative decrease in RBCs and hemo concentration, so the plasma decreases. Then you have, hemodilution would be relative increase and hemo concentration would be relative decrease. Then physiological, so physiological changes in count, as I explained that, altitude or even exercise once you start exercising, the body starts preparing for it and it increases the number of RBC in our body, like it starts producing it. So that's a physiological reason for for having more RBCs. And then erythrocytopenia, high PO2. Okay, so when you're in an oxygen pure environment and there is too much oxygen, the body doesn't want to cause oxygen poisoning to the tissues. That's the reason it reduces the number of RBCs. So if there are lesser RBCs and they're carrying a lot of oxygen, still the tissues won't get poisoned by it. Then we have the erythrocyte size. So there is a really important graph that comes up here. It's a very simple graph. I don't know why it's important, but it is. So we have the Price-Jones curve, which shows microcytes, anisocytosis, and macrocytes. So anisocytosis is basically the normal size. The normal size is 7.2 plus minus 0.5 micrometers. So that is your normal size. And if you shift to the left, then you get microcytes. So microcytes would be like they might ask you a question in the credit. When do you see microcytes? So like it could be because of iron deficiency. Since iron deficient, if iron is deficient, hemoglobin cannot be made. So functional RBCs cannot be made. And iron is also required for the maturation of RBC. So you get microcytes in that case. Or macrocytes. Macrocytes can be in vitamin B12 deficiency or folate deficiency. There's a disease called pernicious anemia or megaloblastic anemia that you can see on the right. Yeah. So that, and then the volume is important, and then reticulocyte count. They might ask you the reticulocyte count. This is the last stage before mature erythrocytes are formed, and they last in the blood for around 1 to 2 days. And basically, they indicate that more RBCs are being formed. So that's just like it's an indicator. So if you have something like blood cancer, then they might check the reticulocyte count. Okay. So then we have hematocrit. Hematocrit is basically the ratio of RBC volume to total blood volume. So hematocrit depends upon size, count, and plasma volume. It depends on size because if you take a drop of blood and you put it under a microscope and you look at RBCs of different size, if the RBCs are really small, then you'll see 3 per unit. But if the RBC is really big, like it's swollen, then you see 1 per unit. So that determines the hematocrit. Then it depends on the count. Obviously, more RBCs, more hematocrit, and then plasma volume. So more plasma volume, lesser the hematocrit because it's a ratio. As you can see, again, men have more hematocrit than women do. So men have more hematocrit because firstly they have more RBCs. Yeah, and then women have less hematocrit because less RBCs, and newborns will have a different hematocrit again because of the RBC change, RBC count change. Yeah. Then there is higher hematocrit in venous blood. So this is really important. So as Aisha explained the carbonic anhydrase system, what happens is only in veins you can find carbon dioxide. The carbon dioxide goes into the RBC and it reacts with water and it forms carbonic acid, which immediately splits into H plus and HCO3 minus. So this happens in tissues and veins. This is because you're picking up the carbon dioxide. And once it splits into H plus and HCO3 minus, H plus can be buffered by the hemoglobin, but HCO3 cannot be buffered. So there's like a huge accumulation of HCO3 inside the RBC, and the HCO3 is forced to be to exit the RBC. So to maintain electroneutrality, so when HCO3 minus leaves your RBC, you're left with a positive charge behind. So to make the RBC electrically neutral, chlorine, as in the Cl minus ion, will enter the RBC and it drags water along with it. So when it drags water along with it, it causes the RBC to swell, and that's the chloride shift. If you guys have any questions in this, please Tell me, I will drop. It's fine? Okay, cool. Then there's higher hematocrit in spleen than kidneys. So there's higher hematocrit in spleen than kidneys because spleen is also known as the RBC graveyard, and it also helps with filtration of blood components. So since there are many RBCs there stuck in the sinusoids, that's the reason the hematocrit is higher in spleen rather than kidneys or something like the liver. It is measured by the microhematocrit method. This is a direct question from the credit. Then the blood volume. So blood volume, guys, you need to learn it. There is no way around it. Blood volume, it is influenced by dehydration, altitude, exercise, and pregnancy. So by dehydration, blood volume will decrease because when you're dehydrated, the water essentially in the blood has decreased, the plasma has decreased. That's the reason the blood volume decreases. Altitude, the blood volume increases. This happens because RBCs, like new RBCs, are made, and also you kind of do feel a little thirsty in like higher altitudes. Then exercise, obviously, again you're feeling thirsty, and RBCs are produced, so the blood volume increases. And then pregnancy, since there's growth and development, just like newborns, you will have more RBCs, and that's the reason higher blood volume. Then the plasma volume, guys, you have to learn the values. Density. Density is basically density is basically dependent on hemoglobin, RBC count, proteins, and water. So you're just looking at how dense the blood is at that specific unit. And if water increases, then the density would decrease. But the rest of the three, hemoglobin, RBC count, or proteins, if they increase, the density will increase too. And viscosity, it's not the same as density. Please don't get confused. Yeah, please don't get confused. So viscosity, it depends on almost the same things: RBC count, proteins, water, temperature, and flow speed. So RBC count, proteins increasing would cause the viscosity to increase because it will make it more dense, but that's not what we're talking about. It's the resistance to flow that we're talking about. If water increases, viscosity decreases. If temperature increases, again viscosity decreases, and same with flow speed. So yeah, that's viscosity. Then the erythrocyte sedimentation rate, which can be denoted as ESR or FW after the method that is used, like Faro-Westerlin method. So that's erythrocyte sedimentation rate. So basically, erythrocyte sedimentation rate is when RBCs they settle when they're left standing for a while. So RBCs, blood is a suspension, and those RBCs settle when it stops flowing. So this is where viscosity becomes important. That's the reason you're supposed to know the value for it. So there are phases of the settling of RBCs. First is aggregation, which is roller formation. Roller formation is basically this coin stacked kind of formation of RBCs. This forms because RBCs have a negative charge, so they have a coat of salicylic acid on them, which is negatively charged, and these RBCs get attached to positively charged plasma proteins, and they form these penny stacked formations, which are roller formations. And the more the roller formations, the more the roller formation, the higher the erythrocyte sedimentation rate. Yeah, the normal values. If you see, men have shorter erythrocyte sedimentation rate, and women have a higher erythrocyte sedimentation rate because women, firstly, they have more plasma proteins and they have lesser RBC count. So if you think about it, if there are less RBCs, and plasma proteins are obviously not going to be as much as the RBCs in our body, if there are less RBCs, then there would be less repulsion between those RBCs, and there would be enough ratio of plasma proteins to RBCs, and they can form rollers. But if there are many RBCs, like in men, then you would have negative electrostatic repulsion, and the roller formation would become slower. So that's the reason females have higher erythrocyte sedimentation rate. Then non-specific marker, yeah. So erythrocyte sedimentation rate is affected by a lot of factors, and since it's affected by a lot of factors, you can only use it to see if there is a plasma protein ratio change, and not really for other things. So it's useful for monitoring diseases but not diagnosing. Okay, so factors affecting erythrocyte sedimentation rate. So as I explained in the earlier slide, RBC count affects the erythrocyte sedimentation rate. Higher the RBC count, the lower the lower the erythrocyte sedimentation rate. Then RBC shape. So when there are abnormal shapes like the sickle shape, which is the drepanocytes or spherocytes, the RBC cannot form rollers easily. That's because the point-stand formation doesn't take place when it's in a different kind of shape, and that's the reason ESR decreases. Then plasma proteins. Okay, so as I explained in the roller formation, we know that the plasma proteins must be positively charged for it to happen. If they're negatively charged, then it's not going to happen. And albumin is one of them. It's like famously negatively charged, and if albumin is high, then the ESR would be low, and vice versa. So that's the reason either the albumin is a plasma protein. If it increases, your ESR is going to decrease. Phospholipids, again, it's the same thing. Then blood composition, so viscosity, lipemia, and hemoglobin concentration also affect ESR. Viscosity could be, if there is more viscosity, that means there is more RBCs, but it depends. If there are more plasma proteins which are positively charged, then the roller formation can increase. So always context matters. Lipemia, either it can be that the lipids they weigh down the RBCs, and that's the reason the sedimentation rate increases, or it can come between the roller formation and it can decrease the erythrocyte sedimentation rate. So again, context matters. Temperature, so higher the temperature, faster the sedimentation, and there are other conditions that are listed below, like malignancy, inflammation, and infection that will cause the erythrocyte sedimentation rate to change. Now, anemia. So anemia is basically the hemoglobin concentration decreasing below the normal limits, and that's the reason oxygen cannot reach your tissues. So the consequences of anemia, again, oxygen cannot reach the tissues, which is tissue hypoxia, changes in hematocrit and viscosity, and fatigue, pallor obviously because the blood is not reaching places, so you have pallor, dyspnea because you don't have oxygen reaching, and weakness, fatigue. Then main causes. So the main causes, there are three types of causes. There's decreased RBC production. So there can be a problem in the bone marrow, which is known as aplastic anemia. Then you can have iron deficiency. Again, iron requires hemoglobin formation and RBC maturation. So if that iron deficiency is there, you can get microcytic anemia. And then megaloblastic anemia, which is because of vitamin B12 or folate deficiency. So when RBCs are maturing, they go from a bigger size to a smaller size because we need the RBCs to go through the capillaries easily. So when you have megaloblastic anemia, they stay in this orthochromatic stage. The orthochromatic stage is basically when the RBCs are really big and they start rupturing. So you get megaloblastic anemia. And then blood loss. Blood loss, again, acute or chronic bleeding, so you're literally just losing RBCs, so you're going to have anemia. And it can be restored in a while. And then you have increased RBC destruction, which is hemolytic anemia. So hemolysis will be explained soon. But hemolytic anemia, I just want you guys to know that megaloblastic anemia, sickle cell anemia, or microcyte formation, They can all lead to hemolytic anemia because our body can just decide, okay, you know what, this is a foreign body and we need to get rid of it, so break them, like rupture, cause them to rupture, and then that's how you get hemolytic anemia.
Okay, so the next topic is going to be hemolysis. And hemolysis is quite a big part of hematology, but we're going to try to break it down to make it as simple as possible. First of all, from the name lysis, it's going to be the destruction or damage to the red blood cell membrane, which is going to cause hemoglobin to be released in the plasma. Once the hemoglobin is released, it's going to lose its oxygen transport function. So basically, it is no longer useful for us. So it can cause toxic effects. For example, kidney damage, hemoglobinemia, all that stuff. But we'll get into that later. But in hemolysis, it can either be physiological or pathological. Physiological because you know that erythrocytes have a 120-day lifespan. So after those 120 days, in the reticular endothelial system, which is a term you need to get familiar with, which is going to be your spleen, liver, and bone marrow, it's going to get phagocytosed, and all the components are going to be recycled, as in iron, globin, and bilirubin. Whereas in pathological hemolysis, premature and excessive destruction of red blood cells in the circulation, intravascular, or in tissues extravascular. Intravascular from the name, this is occurring in the blood vessel itself. In tissues is extravascular, it's occurring outside. This could potentially cause hemolytic anemia if the red blood cell destruction exceeds the production. Hemolysis can occur due to different reasons, but the main thing in common is they are all causing destruction to the red blood cell membrane. The different types is through osmotic stress. We're going to get into the details in the next slide. Mechanical forces, chemical agents, biological and toxic agents, and then immunological reactions. Yeah, so first you have osmotic hemolysis. It's based on the osmolarity and the tonicity of the surrounding fluid. This is a piece of information that you do need to know for the credit and for your final. In an isotonic solution, which is 0.9% NaCl, the red blood cells maintain their normal shape and volume. This you have to know. And then in hypotonic solution, the water is going to enter the cell, causing swelling and then a rupture. And then hypertonic solution, the water is going to leave the cell, causing shrinkage, or the term that professors would prefer you to use is crenation, so there is no lysis happening. And this is used experimentally to determine the osmotic resistance of erythrocytes. The normal resistance, red blood cells begin to hemolyze at 0.45, 0.5% NaCl, and complete hemolysis occurs at 0.3, 0.25. This is also another piece of information that you do need to know. And it's the most important type of hemolysis out of the 5. The second one is physical hemolysis. This is not really needed for your credit, but it's basically caused from some sort of trauma. Needles, valves, shaking. It can also be from temperature, extreme heat or cold, ultrasounds, and shear stress. And then you have chemical hemolysis, also caused by some sort of solvent like chloroform and ether, detergents, acids, bases, oxidizing agents that are basically going to be damaging the lipid bilayer. But again, this is not really important for your credit. And then you have your toxic hemolysis by toxins or poisons like Clostridium, snake venom, heavy metals. They're either going to disrupt the membrane directly or they're going to cause oxidative damage to hemoglobin. And then you have immunological hemolysis, which is, I would also say, important to know. It occurs when the antibodies are going to bind to the antigens on the red blood cell membrane, activating complement system, leading to membrane lysis. I don't believe you guys took complement system in detail yet, but you know that it's an immune response that is basically going to end in some sort of phagocytosis or engulfing of the cell. And the example of this is incompatible blood transfusion, which you guys should have taken by now. And then we have laboratory indicators of hemolysis, things you would expect to find in a sample. First, the plasma color is going to be deeper, more red, because we know that hemoglobin is being released into the plasma, giving us hemoglobinemia. Second thing, urine. You're going to have hemoglobinuria, where you're finding the hemoglobin in the urine, and this is a sign of pathology. And then you have your serum bilirubin. It's going to be increased due to the increase in unconjugated bilirubin, and this occurs extravascularly. We'll get more details in a second. Reticulocyte count is going to be increased to compensate for erythropoiesis because we are losing red blood cells. And then you have haptoglobin, which we'll also get into in the next slide. It's being decreased because it's binding to the free hemoglobin in the plasma. It's being taken up. Okay, so for the hemolytic pathway, we said that it's broken down into intravascular and extravascular, but first we're going to do the entire process all together and then divide it and break it down into intra and extra. This image is found in your final notes. It's really good, and I suggest studying it very well because it summarizes everything. So first, you have your red blood cell, and the hemoglobin is released and split into dimers, as you can see at the top. These dimers are going to bind to haptoglobin, which we already mentioned. It's a plasma protein, and this complex gets phagocytosed and prevents hemoglobin loss in urine, which we mentioned before. Or it has two different fates. The second option is that it's going to get oxidized by nitric oxide, which we know is released by the endothelium, to methemoglobin, which may cause it to disintegrate into globin and heme. However, heme by itself causes oxidative stress. So what we do to compensate for that is bind it to hemopexin, which is eventually going to be absorbed by hepatocytes and degraded to conjugated bilirubin. This is where we sort of entered the extravascular, because we are no longer in blood vessels, we are in hepatocytes. Some of the conjugated bilirubin is going to stay in the large intestine and is metabolized by colonic bacteria to stercobilinogen and then oxidized to stercobilin, which in simple terms is your stool. And some is reabsorbed and excreted in the urine as urobilinogen and the oxidized form, urobilin. And this is basically your urine. And this has to pass by the kidneys at one point. And as you can see in that image over there, stercobilin goes into the kidneys, where you're going to have urobilinogen, gets oxidized urobilin, which is basically your urine. And that is the general hemolytic pathway. But then to specifically break it down into intravascular and extravascular. In intravascular, like we already said, the first step is going to be the red blood cell rupture within the circulation. It could be due to an autoimmune disease like hemolytic anemia, or because of the other examples that we mentioned, like mechanical or chemical and all that stuff. And then the hemoglobin is going to get released into the plasma. It's going to bind to haptoglobin, or like we said, could be oxidized by nitric oxide. And then it's going to be cleared by macrophages. As the haptoglobin gets consumed, it's going to decrease the haptoglobin in plasma. And that's another lab indicator that we already mentioned. When haptoglobin gets depleted, there's going to be extra hemoglobin in the plasma. This gets filtered by the kidneys, could potentially cause hemoglobinuria and hemosideruria. This is a little bit advanced for the pilot, but it's important to know. And then nitric oxide can again play a role causing vasoconstriction, hypertension, and stuff we already have mentioned. But then when we're talking about the extravascular hemolytic pathway, initially it starts by some sort of abnormal red blood cells recognized by macrophages in the reticular endothelial system, like we said, against spleen, liver, bone marrow. And then there's going to be phagocytosis of red blood cells by the macrophages. The red blood cells are broken down inside the phagolysosomes, and then hemoglobin gets degraded into globin, which is going to be going to break down further into amino acids, which are going to get reused. Heme, which is going to give you iron, which is further broken down into your protoporphyrin ring. Iron is going to get recycled via transferrin, and then the protoporphyrin ring is going to turn into biliverdin, and then into bilirubin, which is the unconjugated form. Bilirubin gets released into plasma, and the unconjugated bilirubin binds to albumin, gets transported to the liver. In the liver, the bilirubin is going to get conjugated with glucuronic acid via glucuronide transferase enzyme, And that's when it's going to be converted into urobilinogen and stercobilinogen, and then gets excreted by the body either in the form of urine or in the form of stool. It's actually very simple. All this needs is constant repetition, and then the concept will be very easy. And that's all.
Okay guys, before we start the last part, I want to ask, have you guys done the experiments in lab? Just one class you had, right? Okay. So you have not done APTT, okay. Okay, no problem. We'll just go over it, we'll summarize it. So I'm happy to tell you that you will be having lab tests also included in your test. So you should be knowing all the theory about it. Yeah. So first you have your bleeding time or Duke method. So here the main principle is you are measuring the clotting time, the platelet activation time. So when a small vessel is injured, the platelets will adhere to the collagen, form the hemostatic plaque, and the time taken for this process is the bleeding time. Now, do you guys want to know how the experiment is done, or do you want to know in lab class? You want to know? Okay. So basically, you'll be piercing your ear, and then and you'll be taking like the what do you call them? Yeah. On a paper, you'll just be taking the blood, what do you call them?
Like you take the clotting paper and
Yeah.
you're gonna put it on the ear where it's like pierced, and you're gonna see how long it takes for the bleeding to stop.
Yeah, you will just keep blotting again. You take the blots of your blood, and you will be like recording how long it takes for the bleeding to stop. That's your platelet formation. So it usually takes 2 to 3 minutes, and there are around 11 to 13 blots which you will see on your paper. So that is your Duke method. That's your bleeding time. Then we have clotting time after Lee and White. So again, it's the same principle. Again, you're measuring the clotting time, but the procedure is a bit different. So you'll be collecting blood using venipuncture, and then you'll be placing that blood in a test tube and keeping it just there for a few minutes to see how long it takes for the blood to clot. And the main principle is that when blood contacts a foreign surface, it will activate the intrinsic clotting pathway, which was explained very much in detail, okay, and lead to the formation of clot. And this usually takes around 10 to 15 minutes. So you're not adding anything, just like letting the blood clot into the test tube. Okay, so this is your clotting time after Lee and White. Then we have very, very important activated partial thromboplastin time, APTT. Okay, so the key words in this, you have to remember. The activator, which is ellagic acid and phospholipid, that is cephalin, it is added into that same venipuncture blood which you collect, and along with calcium to trigger the clotting. Okay, so in this procedure as well, you collect the blood in a test tube, and you add these substances, and you just wait for the blood to clot. And it usually takes around 30 to 40 seconds. So that's what you'll be measuring in APTT. And lastly, you have prothrombin time test. This is the quick test. So it's again the same procedure. You'll be adding some substances. Here is the tissue factor and phospholipids and calcium ions will be added to the plasma. So it's a bit different from APTT in that sense. And you will again be seeing the clot formation, that is the prothrombin time. Okay, and this test is shorter. It takes around 13 to 14 seconds. Okay, so these are your main tests. I think you did erythrocyte sedimentation, and yeah, those are also important, but you must know the theory for this because you may get any question. It took us by surprise as well last year when we got it.
We weren't told how important these tests are, and then when they came up in our credits, we were like pretty shocked.
Yeah, so we are telling it to you in advance, so remember to go through them, okay? And I think that brings us to the end of our seminar. So yes, it was very long, it was very detailed. We did around 3-4 lectures in one seminar. So yeah, do you have any questions? Up till now?
None of you started studying, right? That's why you're confused. But once you start studying and you understand everything, you can always reach out to us with any questions.
enjoy to like understand the
Yeah.
topics, but then like learn from the lecture.
Yeah.
That's very, very important. You have to
You'll
learn
get
from
your
the
like
lectures.
good basics down with internet, but obviously whatever's in the lecture is the more important.
Yeah. You can use textbook as well, but I feel like they're way more
Yeah.
detailed.
Guided
than-
like
Mm.
I don't
not
think any of us use guided
Yeah.
analysis.
I would not recommend that much. You can use final notes as well if you want to
Well,
get an understanding, but main
Yeah.
resource should be the lecture, okay? So like study from the lecture very well. And apart from that, yeah, and these lab tests also.
Yeah.
Study the theory very well, yeah?
This is from the materials they give for lab class.